Hello, welcome to my College Scholars Capstone presentation. Thank you for your interest in my project. My project is called Cis eQTL analysis in the HMDP hypothalamus dataset. Now, if you're a little new to genetics and bioinformatics, you may have no idea what this means at all. Don't worry, I'll build up enough background information for you to follow along as I explain my project. For those well informed on this topic, feel free to skip ahead if what I present is redundant. In this presentation, I'll first present some background information on my research topic. Then I'll go over my methods and results, what I'm currently working on and will work on, and I'll conclude with some acknowledgements. I'd like to share first a little bit about myself and why I chose to do my project on this topic. My name is Justin Yi, and I'm a fourth-year student at UCLA. My major is mathematics of computation. It's a mix between math and computer science with a bit more emphasis on math. I'm also pursuing a minor in Spanish. I entered UCLA interested in pursuing a career in medicine as a physician. But by taking computer science classes my first year, I realized that I also really enjoyed coding. I became an undergraduate researcher for the Xia Yang Bioinformatics Lab in my second year. It is with this lab that I conduct my project. This capstone integrates my academic interests, mathematics, programming, and medicine, together into one challenging project. It has also given me the opportunity to work on my scientific communication skills as I present my work to the public. Alrighty, on to some background information. First, I'd like to begin with the big picture. How will my results be helpful to science and society? Well, heart disease and stroke are still the leading causes of death for men and women in the US. Additionally, in the last 20 years, the number of adults diagnosed with diabetes has more than doubled as the American population has aged and become more overweight and obese. Almost all the top health problems of the Western world are related to cardiometabolism. This is where my study applies. By studying the genetic mechanisms that influence these disorders, we can find genes and biological pathways to target and improve treatment. We can also better predict and characterize patients' health outcomes and how they respond to the prescribed medicine. All right, now onto the big question. What in the world is Cis eQTL analysis in the HMDP hypothalamus dataset? Let's begin with Cis eQTLs. Our DNA is extremely similar to each other, but there are differences. One source of difference is the single nucleotide polymorphism, or SNP. A SNP is a change at a single base position in DNA. Now, an eQTL is an acronym that stands for expression quantitative trait loci. eQTLs are SNPs that are correlated with the differences in gene expression levels. That means that the SNP is correlated with the amount of RNA or protein that a gene makes. An eQTL is a cis eQTL if it is close to a gene. Close means different things, but usually the SNP has to be on the same chromosome and within 1 million base pairs of the gene. Let's analyze this figure on the right of the slide. We have SNP X, which is a cis eQTL because it affects the gene expression levels of gene A, and SNP X is located close. Since humans have two chromosomes, for example, AT on one chromosome, GC on the other, or GC, there's three different combinations for the SNP X genotype. The y-axis shows how much RNA and protein is made by gene A based on which genotype the person has. Each dot is a sample, so we can see by this curved line that the genotype is correlated with gene expression levels. HMDP stands for Hybrid Mouse Diversity Panel. A description of it is written on the slide, but for a brief explanation, mice are a common organism used to model human diseases. Scientists have inbred strains of mice in such a way that each strain exhibits traits of common diseases like obesity, osteoporosis, atherosclerosis, etc. Our data is the single-cell RNA sequencing data from the cells of the hypothalamus of HMDP mice. The hypothalamus is the region of the brain that coordinates our homeostasis. Thus, it coordinates hunger, thirst, body temperature, and other regulatory functions as seen as this figure. Single-cell RNA sequencing is used to get the gene expression levels and genetic code of each gene expressed in single cells. It's an advancement from bulk RNA sequencing, which gets the same information, but from tissue and organ samples, not individual cells. So to recap, what is Cis eQTL analysis in the HMDP hypothalamus dataset? It is finding Cis eQTLs, the changes in the single base position of our DNA sequence that correlate with different gene expression levels. Why is Cis eQTL analysis important? We may find changes in the DNA sequence that influence how certain genes work. These genes may play an important role in diseases and disorders related to the hypothalamus. This helps scientists find targets for future medical treatments. I'll now describe my research project in detail. I went over the first project goal in my background slides. The second goal of the project is to identify Cis eQTLs that overlap with GWAS genes that play a role in cardiometabolic traits and diseases. What this means is that there are other studies called GWAS or genome-wide association studies that identify genes associated with a particular disease. Usually what happens in GWAS and Cis eQTL studies is that thousands, if not tens of thousands, of candidate genes are found. We want to find the overlap between these genes so we can narrow down the list of candidate genes to investigate further in resource-intensive wet lab experiments. The data used in this project is the following. We have an annotated gene expression counts for 29 mice strains, the list of SNPs and their locations in the DNA sequence, and a list of genes and their locations. The table on this slide is a simplified version of the annotated gene expression data that I work with. So each entry shows the gene expression levels given a gene and a mice strain. Remember that each strain of mouse is bred to exhibit different physical traits, so we would expect to see different gene expression levels for different genes among the different strains. The plot on this slide visualizes the different cell types that we are working with within the hypothalamus. They are color classified based on which genes are expressed. And as the lab continues to study the data set, they update each sample's data set. So initially we started with this, but then the lab updated the data set to be a little bit more specific and accurate. To find Cis eQTLs, we did the following. We pre-processed the data, selected a model for detecting Cis eQTLs, evaluated the results, and analyzed the results. When necessary, we backtracked based on feedback from experienced researchers in this field. My work involves writing Python, R, and shell scripting code to manipulate data for analysis and results processing. I mainly use Python software libraries like Pandas, NumPy, and ScanPy. And I also had to learn how to pass the data into higher level analysis algorithms like matrix eQTL and FastLMM. At the end of my project, my lab will receive the hypothalamus Cis eQTLs, as well as a pipeline for detecting Cis eQTLs for any new data sets that the lab may have. The code segment pasted on this slide shows how we pre-process the data. First, the gene counts needed to be separated by cell type because we're trying to find cell type specific eQTLs. Afterwards, we removed the genes with no expression, those not expressed in less than 25 strains, and we removed the strains with no cells present. We averaged the gene expression of each gene for each strain to reduce certain strains that had more cell samples from dominating the analysis. The algorithms we used to find Cis eQTLs required the data to follow a normal distribution, so we quantile normalized and z-scored. Our data was created in 3 separate experimental batches. When this happens, differences in how each experiment is run can affect the data in a way that is related purely to the experiment and not biology. We correct this by finding the covariates of the data. Probabilistic estimation of peer residuals, peer, is a software package that finds these hidden factors. We use the peer factor results as covariates to correct for batch effects. This first graph about batches is a UMAP projection to show how the data is largely separated by batches, as there's not a lot of overlap between these colors. This other graph plots the first and second pure factors and colors each point by batch. V1 is the first pure factor, and we can see that it linearly separates the data by batches for the most part. This means that this first pure factor could be used as a potential covariate. Our first model for finding Cis eQTLs was a linear regression model. Matrix eQTL is an R package that was designed to be more computationally efficient in detecting eQTLs. For a simplified version of how this model works, consider this graph once more. Each dot is a sample from a different strain, and linear regression is finding this line of best fit. And SNP-X is an eQTL if this line of best fit is statistically significant. For our results, I found 12,101 significant Cis eQTLs that overlapped with those in GWAS studies and 52,287 that did not. These figures are box plots that show an eQTL overlapped with the GWAS adiposity or obesity trait, and each dot belongs to a different strain of mice. And we see that genotype is correlated with gene expression levels. These tables summarize some important numbers. On the left, we have the number of genes that have an eQTL associated with it in each cell type. On the right, we have the number of Cis eQTLs present for each cell type. We see that neuron has the most amount of eQTLs compared to the other cells. This may be partially due to the high number of neuron cells sampled in this experiment. One last results slide is a heat map. This is used to visualize the number of eQTLs given a GWAS trait on this x-axis and a cell type on the y-axis. The darker the box, the more eQTLs there are. If we look only at these cell types, we can see that neurons generally have the most eQTLs present for all the GWAS traits. If we look at the GWAS traits, adiposity or obesity seems to have the most amount of eQTLs associated with it amongst all cell types. To look at a specific example, we can look at MCHC, or the average amount of hemoglobin in our red blood cells. It looks like ependymal, oligodendrocyte, and neuronal cells have a high number of eQTLs associated with this MCHC trait. The second model I worked with is called a linear mixed model. Linear mixed models build upon the linear regression and are used to analyze data that is non-independent. Our data comes from inbred strains of mice, so population structure may make the data non-independent. Population structure is the presence of genetic variants within subgroups and is caused by non-random mating. This causes problems for data analysis because a genetic variant that is common in a population with a higher incidence of a trait may be purely coincidental. That means data analysis that doesn't correct for this will incorrectly associate this variant with the disease. Our second model does correct for this by linking a genetic relationship matrix with the linear mixed models. And that is what I'm currently working on right now. I'm using the FastLMM algorithm to find more meaningful Cis eQTLs in our dataset. Additionally, I'm performing a couple of tests to verify the integrity of our results. One test is called permutation analysis, where I'll switch the strain labels to disassociate the genes from the strain. This means that if we find any Cis eQTLs from this data, those Cis eQTLs are false discoveries and due to random chance. We'll use this permutation test to also find a p-value cutoff that will minimize the false discovery rate. After this project is over, we can conduct further research into the genes that we have found. If there are any genes that stand out, we can conduct laboratory experiments to see if the SNPs are the causes of differences in gene expression and not just purely correlated. Gene network analysis, which investigates how genes interact with each other, is also another future project. I would like to first acknowledge and thank the Xia Yang Lab for the opportunity to work on this project by providing me with their data and guidance. Specifically, I would like to thank Dr. Yang and my mentor, Russell Lippman, for teaching me how to do this research and guiding me through the steps of this project. I would also like to thank the College Scholars Program for motivating me to do this project through their personal, academic, and professional development opportunities offered through their interdisciplinary program design, counselor mentorship, and cohort meetings. Here are the references used in this presentation. And if you have any further questions about me or my research, you can reach out to me through my email and I will be happy to talk. Thank you so much.